Enterobacter cloacae, Wikipedia audio. EC subsp cloacae, EC subsp dissolvens. Bacillus cloacae Jordan 1890, Bacterium cloacae Lehmann and Newman 1896, Cloacae Castellani and Chalmers 1919, Enterobacter cloacae Bergi et al. 1923, Enterobacter cloacae Hormici and Edwards 1958, Erwinia dissolvens Burkholder 1948, Pseudomonas dissolvens Rosen 1922, Bacterium dissolvens Rosen 1922, Phytomonas dissolvens Rosen 1926, Aplanobacter dissolvens Rosen 1926, Enterobacter dissolvens Waldi 1945, Enterobacter dissolvens Brenner et al. 1988. Enterobacter cloacae is a clinically significant gram-negative, facultatively anaerobic, rod-shaped bacterium. Microbiology. In microbiology labs, E. cloacae is frequently grown at 30 degrees Celsius on nutrient agar or broth or at 35 degrees Celsius in tryptic soy broth. It is a rod-shaped, gram-negative bacterium, is facultatively anaerobic, and bears Peritrichous flagella. It is oxidase negative and catalase positive. Enterobacter cloacae has been used in a bioreactor-based method for the biodegradation of explosives and in the biological control of plant diseases. E. cloacae is considered a biosafety Level 1 organism in the United States and Level 2 in Canada. A draft genome sequence of Enterobacter cloacae subsp cloacae was announced in 2012. The bacteria used in the study were isolated from giant panda feces. Enterobacter cloacae is a member of the normal gut flora of many humans and is not usually a primary pathogen. Some strains have been associated with urinary tract and respiratory tract infections in immunocompromised individuals. Treatment with cefepime and gentamicin has been reported. A 2012 study in which Enterobacter cloacae transplanted into previously germ-free mice resulted in increased obesity when compared with germ-free mice fed an identical diet, suggesting a link between obesity and the presence of Enterobacter gut flora. E. cloacae was described for the first time in 1890 by Jordan as Bacillus cloacae, and then underwent numerous taxonomical changes, becoming Bacterium cloacae in 1896, cloacae in 1919, it was identified as Enterobacter cloacae in 1923, Enterobacter cloacae in 1958 and E. cloacae in 1960, by which it is still known today. E. cloacae is ubiquitous in terrestrial and aquatic environments. These strains occur as commensal microflora in the intestinal tracts of humans and animals and play an important role as pathogens in plants and insects. This diversity of habitats is mirrored by the genetic variety of the nomen species E. cloacae. E. cloacae is also an important nosocomial pathogen responsible for bacteremia and lower respiratory tract, urinary tract, and intra-abdominal infections, as well as endocarditis, septic arthritis, osteomyelitis, and skin and soft tissue infections. The skin and the GI tract are the most common sites through which E. cloacae can be contracted. E. cloacae tends to contaminate various medical, intravenous and other hospital devices. Nosocomial outbreaks have also been associated with colonization of certain surgical equipment and operative cleaning solutions. Another potential reservoir for nosocomial bacteremia is the heparin solution used to irrigate certain intravascular devices continually. This fluid had been implicated as a reservoir for outbreaks of device-associated bacteremia in several instances. In recent years, E. cloacae has emerged as one of the most commonly found nosocomial pathogen in neonatal units, with several outbreaks of infection being reported. In 1998, Van Nierop et al. Reported an outbreak in a neonatal intensive care unit with 9 deaths, and in 2003, Kuboyama et al. Reported 3 outbreaks with 42 systemic infections and a mortality of 34%. This microorganism may be transmitted to neonates through contaminated intravenous fluids, total parenteral nutrition solutions and medical equipment. Many single clone outbreaks, probably caused by cross-transmission via healthcare workers, have been described, suggesting that inpatients can also act as a reservoir. The type strains of the species are E. cloacae ATCC 49162 and 13047. This latter strain is the first complete genome sequence of the E. cloacae species, and the type strain is E. cloacae subsp. cloacae. Industrial use. The complete E. cloacae subsp. cloacae ATCC 13047 genome contains a single circular chromosome of 5,314,588 BP and 2 circular plasmids, PECLA and PECLB, of 200,370 and 85,650 BP. The other genomes of E. cloacae that have been sequenced are deposited in GenBank under accession numbers CP002272, CP002886, FP929040, and AGSY00000000. E. aspergii is named after Mary Alice Fife Esbury, an American bacteriologist who made many important contributions to the classification of Enterobacteriaceae, particularly in describing new Klebsiella and Salmonella serotypes, new genera, and new species. E. aspergii sp. November was described in 1986 based on the enteric group 17. This group was defined in 1978 as a group of biochemically similar strains isolated from different human specimens and sent to the CDC. Before the designation of enteric group 17, these strains had been reported as unidentified or atypical Citrobacter or Enterobacter strains. After several studies, it was shown that these strains represent a single new species in the genus Enterobacter, which was named E. aspergii. E. aspergii strains have been isolated from the soil and implicated in the mobilization of phosphate for plant nutrition from calcium phosphate, but most E. aspergii species have been isolated from human sources. The type strain of the species E. aspergii is ATCC 35953 and was isolated from lochia exudates of a 22-year-old woman in the USA. The only sequenced strain of E. aspergii is LOF7A, which contains a circular DNA and 2 circular plasmids, Pentis 01 and Pentis 02, which were submitted by Lucas et al. In 2011 to the U.S. DOE Joint Genome Institute. E. hormichii is named after S. Neo Hormichi, a Uruguayan microbiologist who proposed and defined the genus Enterobacter. The name E. hormichii was formerly called Enteric Group 75, which contained 11 strains that were sent to the CDC for identification between 1973 and 1984. 12 additional strains were received from 1985 to 1987, 3 of which were blood isolates. E. hormichii was first described on the basis of 23 isolates sent to the CDC for identification. At that time, they could not be assigned to a species since they were negative in the D-sorbitol and Melibiose tests and did not fit the biochemical profile of any established Enterobacter species. The species E. hormicii was proposed to be lactose, D-sorbitol, raffinose, Melibiose and esculin negative and 87% dulcitol positive. These species were originally defined by O'Hara et al. When a large hybridization group of enteric organisms was isolated and found to be associated with bloodstream infections. The type strain of E. hormicii is ATCC 49162 and was isolated from the sputum of a man in California in 1977. The whole genome shotgun sequencing project was submitted in 2011 to the Human Genome Sequencing Center. E. hormicii consists of 3 different subspecies: E. hormicii subsp. O'Hara, E. hormicii subsp. hormicii and E. hormicii subsp. Steiger-Waltei, which corresponds to genetic clusters 6, 7, and 8, respectively. The differentiation of these subspecies is based on their particular properties and biochemical tests. Safety. Genomics. E. hormicii is commonly isolated as a nosocomial pathogen of clinical significance. It has been reported in several outbreaks of sepsis in neonatal intensive care units in the USA and in Brazil, where the outbreak originated from contaminated parenteral nutrition. Clinical significance. Species of the E. cloacae complex. E. Kobe is named after Kobe City, where the type strain of this species was isolated. E. Kobe was first described by Kosako et al. Based on a collection of 23 strains with the general traits of E. cloacae and the common phenotypic difference of being Voges Proskauer negative. The name E. Kobe is proposed for a group of organisms referred to as NIH Group 21 at the NIH, Tokyo. It was later found that NIH Group 21 also resembled the CDC enteric Group 69, and E. Kobe was compared with the latter. On the basis of DNA relatedness, both organisms could be included in a single taxon. However, the CDC enteric Group 69 was described as positive in Voges Proskauer and yellow pigmentation, whereas all strains of E. Kobe were Voges Proskauer and pigmentation negative. These findings suggest that the relationship of both organisms is at the subspecies or biogroup level. The type strain of E. Kobe is NIH 1485-1479 and was isolated by blood culture of a diabetic patient. E. ludwigii, named after Wolfgang Ludwig, a microbiologist working in bacterial systematics and who developed the ARB databases as well as making them public. This description is based on the phylogenetic analyses of partial HSP60 sequence data collected in a population genetic study, as well as on DNA hybridization assays and phenotypic characterizations. The type strain N119T was isolated from midstream urine of an 18-year-old male patient with a nosocomial urinary tract infection while he was hospitalized at the Großadern University Hospital Munich, Germany. The GenBank accession number of the 16S rDNA of strain N119T is AJ853891. E. nimipressuralis. The species E. nimipressuralis was originally defined by Brenner et al. And was formerly called Erwinia nimipressuralis, which was isolated from non-clinical sources. Erwinia nimipressuralis was inserted in the approved lists of bacterial names in 1980. This microorganism is biochemically similar to E. cloacae, but it is different for acid production from sucrose and raffinose, whereas E. cloacae is positive in these tests. The type strain of E. pressuralis is ATCC 9912 and isolated from the L. mulmus species in the USA. E. cloacae subsp. cloacae strain PR4 was isolated and identified by 16S rDNA gene sequence with phylogenetic tree view from explosive-laden soil by P. Ravi Kumar. E. cloacae SG208 identified as a predominant microorganism in mixed culture isolated from petrochemical sludge, IOCL. Guwahati is responsible for degradation of benzene was reported by Patty and Gokula. 1. Sanders W. Jr., Sanders C.C. Enterobacter species, pathogens poised to flourish at the turn of the century. Clin. Microbial. Revelation 10,22241. Exhaustive review on the Enterobacter genus that highlights microbiological, clinical, and epidemiological features and antibiotic susceptibility. 2. Streit J.M., Jones R.N., Sader H.S., Fritsch T.R. Assessment of pathogen occurrences and resistance profiles among infected patients in the intensive care unit. Report from the Sentry Antimicrobial Surveillance Program. INTJ Antimicrobe. Agents 24,111-118. 3. Hydron AI, Edwards Jr., Patel Jetal, for the National Healthcare Safety Network team and participating National Healthcare Safety Network facilities. Antimicrobial resistant pathogens associated with healthcare-associated infections. Annual summary of data reported to the National Healthcare Safety Network at the Centers for Disease Control and Prevention, 2006-2007. Infect. Control HOSP. Epidemial point 29,996-1011. 4. Wisplinghoff H., Bischoff T., Talent SMETL. Nosocomial bloodstream infections in US hospitals, analysis of 24,179 cases from a prospective nationwide surveillance study. Clin. Infect. DIS 39,309-317. 5. PAE, Caspers MP, Skuren FHETL. Genomic diversity within the Enterobacter cloacae complex. +1 3E3018. Utilization of 4 genetic approaches for discriminating within the Enterobacter cloacae complex. 6. Hoffman H., Roggenkamp A. Population genetics of the nomen species Enterobacter cloacae. APPL. Environ. Microbial point 69,5306-5318. Genetic clustering highlighting taxonomic and epidemiological characteristics of the E. cloacae complex. 7. Hormichi E., Edwards Pr. A proposed genus Enterobacter. Int. Vol. Bacterial. Nomenclature. Taxon point 10,7174. 8. Morand P.C., Billowit A., Rotman METL. Specific distribution within the Enterobacter cloacae complex of strains isolated from infected orthopedic implants. J. Clin. Microbial point 47,2489-2495. 9. Wang Gf, Xie Gl, Zhu BETL. Identification and characterization of the Enterobacter complex causing mulberry wilt disease in China. EURJ plant pathol point 126,465-478. 10. O'Hara CM, Steigerwald AG, Hill BC, Farmer JJ3, Fanning GG, Brenner DJ. Enterobacter hormicii, a new species of the family Enterobacteriaceae formerly known as enteric group 75. J. Klin. Microbial. 27, 2046 2049. 11. Hoffman H., Stindl S., Ludwig W. et al. Enterobacter hormicii subsp. Harry subsp. November, E. hormicii subsp. hormicii comb. November, and E. hormicii subsp. Steiger Walti E. subsp. November. 3 new subspecies of clinical importance. J. Klin. Microbial. 43 32 97 33 03. 12. Hoffman H., Stindl S., Stumpf et al. Description of Enterobacter ludwigii sp. November, a novel Enterobacter species of clinical relevance. SYST. APPL. Microbial. 28, 206 212. 13. Hoffman H., Stindl S., Ludwig W. et al. Reassignment of Enterobacter dissolvens to Enterobacter cloacae as E. cloacae subspecies dissolvens comb. November. An amended description of Enterobacter aspergii and Enterobacter cobae. SYST. APPL. Microbial. 28, 196 205. 14. Mishana S.E., Jerwing L., Mind MET.L. Outbreak of a novel Enterobacter sp. carrying BLA CTXM15 in a neonatal unit of a tertiary care hospital in Tanzania. INTJ antimicrobe. Agents. 38, 265 269. 15. Pavlovic M., Conrad R., Iwabian, Singh A., Bush U., Huber I. A dual approach employing MALDI-TOFMS and real-time PCR for fast species identification within the Enterobacter cloacae complex. FEMS microbial. Let. 328, 4653. 16. Hoffman H., Schmold S., TRQLZSCHK et Nosocomial urosepsis caused by Enterobacter cobae with aberrant phenotype. Dian. Microbial. Infect. DIS 53, 143 147. 17. Townsend S.M., Hurl E., Kabila J., L.O.C. Carrillo C., Forsyth S.J. Characterization of an extended-spectrum beta-lactamase Enterobacter hormicii nosocomial outbreak, and other Enterobacter hormicii misidentified as Chronobacter sakazaki. Microbiology 154 36 59 36 67. 18. Guerra Izar J., Kaufman Me, Pitt T.L. Comparison of ribodyping with conventional methods for the type identification of Enterobacter cloacae. J. Klin. Microbial. 29, 1303 1307. 19. Hartel R., Bandlow G. Epidemiological fingerprinting of Enterobacter cloacae by small fragment restriction endonuclease analysis and pulsed field gel electrophoresis of genomic restriction fragments. J. Klin. Microbial. 31, 128 133. 20. Williams J. G. K., Kubalik A. R., Livak K. J., Rafalski J. A., Tinggi S.V. DNA polymorphisms amplified by arbitrary primers are useful genetic markers. Nucleic acids RES 18, 65 31 65 35. 21. Stumpfen, Roggenkamp A. Hoffman H. Specificity of enterobacterial repetitive intergenic consensus and repetitive extragenic palindromic polymerase chain reaction for the detection of clonality within the Enterobacter cloacae complex. Dian. Microbial. Infect. DIS 53,916. 22. Barnes AI, Ortiz C., Parage M.G., Balin Zeno L.E., Albisa I. Purification and characterization of a cytotoxin from Enterobacter cloacae. Ken J. Microbial. 43,729 733. 23. Stuber K., Frey J., Bernans A. P., Cunard P. Detection of type 3 secretion genes as a general indicator of bacterial virulence. Mole. Cell. Probe 17,2532. 24. Krzyminska S., Mokraka J., Kachura R., Kaznowski A. Cytotoxic activity of Enterobacter cloacae human isolates. FEMS Immunol. Med. Microbial. 56,248 252. 25. Krzyminska S., Kachura R., Mokraka J., Puton T., Kaznowski A. Isolates of the Enterobacter cloacae complex induce apoptosis of human intestinal epithelial cells. Microbe. Pathog. 49,8389. 26. Olsen A., Arnqvist A., Hammer M., Normark S. Environmental regulation of curly production in Escherichia coli. Infect. Agents DIS 2,272 274. 27. Zogaj X., Bacrons W., Nims M., Romling U. Production of cellulose and curly fimbriae by members of the family Enterobacteriaceae isolated from the human gastrointestinal tract. Infect. Immune. 71,41 51 41 58. 28. Kim S.M., Lee H.W., Choi Y.W. et.al. Involvement of curly fimbriae in the biofilm formation of Enterobacter cloacae. J. Microbial. 50, 175 178. 29. Lee So, Kim Y.S., Kim B.N., Kim M.N., Wu J.H., Ryu J. Impact of previous use of antibiotics on development of resistance to extended-spectrum cephalosporins in patients with Enterobacter bacteremia. EURJ Clin. Microbial. Infect. DIS 8,577 581. 30. Musil I., Jensen V., Schilling J., Ashdown B., Kent T. Enterobacter cloacae infection of an expanded polytetrafluoroethylene femoral popliteal bypass graft, a case report. J. Med. Case Republic 9,131. 31. Dalban M., Varkalgia G., Basso M. et.al. Investigation of an outbreak of Enterobacter cloacae in a neonatal unit and review of the literature. J. Hosp. Infect. 70, 714. 32. Van Nierop W.H., Duze A.G., Stewart R.G., Bilger Reyer, Kornhoff H.J. Molecular epidemiology of an outbreak of Enterobacter cloacae in the neonatal intensive care unit of a provincial hospital in Gauteng, South Africa. J. Clin. Microbial. 36 30 85 30 87. 33. Kuboyama R.H., De Oliveira H.B., Moretti Branchini M.L. Molecular epidemiology of systemic infection caused by Enterobacter cloacae in a high-risk neonatal intensive care unit. Infect. Control HOSP. Epidemial point 24,49494. 34. Ren Y, Josie et al. Complete genome sequence of Enterobacter cloacae subsp. Cloacae type strain ATCC 13047. J. Bacterial point 192,2463-2464. 35. Edwards PR, 5 Ma. Capsular types of Klebsiella. J. Infect. DIS 91,92104. 36. Edwards PR, 5 MS. 11. Undescribed Arizona serotypes isolated from man. Anthony van Leeuwenhoek 28,402404. 37.5 Ma, McWhorter AC, Edwards PR. 10. New Arizona serotypes isolated from animals and animal food products. Anthony van Leeuwenhoek 28,369-372. 38. Manzano D, Rojo P, Zubro Z, Alvarez M, Santa Maria J M, Cisterna RN Firm. Infec. Microbial. Clin.9,186-187. 39. Farmer J.J. 3rd, Fanning Gr., Davis Br. et al. Escherichia fergusonii and Enterobacter taylori, 2 new species of Enterobacteriaceae isolated from clinical specimens. J. Clin. Microbial point 21,7781. 40. Ewing WH, 5 Ma. Enterobacter agglomerans comb. November. INTJSYST. Bacterial point 22,411. 41. Farmer JJ 3, Asbury Ma, Hickman FW, Brenner DJ, Enterobacteriaceae Study Group. Enterobacter sakazaki, a new species of Enterobacteriaceae isolated from clinical specimens. INTJSYST. Bacterial point 30,569-584. 42. Grimont Pad, Grimont F, Farmer JJ3, Asbury Ma. Cetacea davisi General November, SP November, and Cetacea lepigii SP November, new Enterobacteriaceae from clinical specimens. INTJSYST. Bacterial. 31,317-326. 43. Brenner DJ, McWhorter AC, Chi A, Steigerwald AG, Farmer JJ3. Enterobacter asbury ESP November, a new species found in clinical specimens, and reassignment of Erwinia dissolvens in Erwinia nimipressuralis to the genus Enterobacter as Enterobacter dissolvens comb. November and Enterobacter nimipressuralis comb. November. J. Clin. Microbial point 23,1114-1120. 44. Farmer JJ3, Davis Br., Hickman Brenner FW et al. Biochemical identification of new species and biogroups of Enterobacteriaceae isolated from clinical specimens. J. Clin. Microbial point 21,4676. 45. Davin Reglier, Bosi C, Charles R. et al. A nosocomial outbreak due to Enterobacter cloacae strains with the E. hormicii genotype in patients treated with fluoroquinolones. J. Clin. Microbial. 35, 1008 1010. 46. Pa. Caspers mpm, Leverstein van Halma et al. Identification of resistance and virulence factors in an epidemic Enterobacter hormicii outbreak strain. Microbiology 155, 1478 1488. 47. Wenger P.N., Talkers G., Brennan P. et al. An outbreak of Enterobacter hormicii infection and colonization in an intensive care nursery. Clin. Infect. DIS 24, 1243 1244. 48. Campos L.C., Lobianco L.F., Saki L.M., Santos R.M., Asensi M.D. Outbreak of Enterobacter hormicii septicemia in newborns caused by contaminated parenteral nutrition in Brazil. J. Hosp. Infect. 66, 95 97. 49. Kosaka Y., Tamura K., Sakazaki R., Miki K. Enterobacter Kobe sp. November, a new species of the family Enterobacteriaceae resembling Enterobacter cloacae. Cur. Microbial. 33, 261 265. 50. Farmer J.J. Enterobacteriaceae. In Manual of Clinical Microbiology. Murray P.R., Baron E.J., Pfaller Ma, Tenover F.C., Yokin R.H. American Society for Microbiology, Washington, D.C., USA, 438-449. 51. Ludwig W., Klenk H.P. Overview, a phylogenetic backbone and taxonomic framework for prokaryotic systematics, in Berge's Manual of Systematic Bacteriology. Garrity G. Springer, New York, USA, 4965. 52. Ludwig W., Strunko, Westrom R. et al. ARB, a software environment for sequence data. Nucleic acids RES 32, 1363 1371. 53. Stockai, Kruger T., Wiedemann B. Natural antibiotic susceptibility of strains of the Enterobacter cloacae complex. INTJ antimicrobe. Agents. 18, 537 545. Evaluation of a wide range of antibiotics tested against E. cloacae, Enterobacter hormicii, and Enterobacter asperii strains, providing a database for their natural susceptibility. 54. Kim D.M., Zhang S.J., Nupane G.P. et al. Enterobacter nimipressuralis as a cause of pseudobacteremia. BMC infect. DIS 10, 315. 55. Scotta C., Wan C., Cabot G. et al. Environmental microbiota represents a natural reservoir for dissemination of clinically relevant metallobeta-lactamases. Antimicrobe. Agents. Chem other. 55, 5376 5379. 56. George A.J. AMP-C beta-lactamases. Clin. Microbial. Revelation 22, 161 182. 57. Ro K.H., Song W., Chung H.S. et al. Chromosomal cephalosporinase in Enterobacter hormicii as an ancestor of ACT1 plasmid-mediated AMP-C beta-lactamase. J.Med. Microbial. 61, 94 100. 58. Choi S.H., Lee J.E., Park S.J.E.T.L. Prevalence, microbiology, and clinical characteristics of extended-spectrum beta-lactamase producing Enterobacter species, Serratia marcescens, Citrobacter freundii, and Morganella morgani in Korea. EURJ Clin. Microbial. Infect. DIS 26,557,561. 59. Smith Molland E., Sanders C.C., Thomson K.C. Kin results obtained with commercially available microscan microdilution panels serve as an indicator of beta-lactamase production among Escherichia coli and Klebsiella isolates with hidden resistance to expanded spectrum cephalosporins and Astrianam. J. Clin. Microbial. 36,2575-2579. 60. Salepi E., Gaiacau pp. Safayana D., Lakova v. Camaragloe, Psacres a detection of extended spectrum beta-lactamases in clinical isolates of Enterobacter cloacae and Enterobacter aerogenes. J. Clin. Microbial. 38,542-546. 61. Psovlakis ls, Vatipolos ac, Katsanis g, Salepi e. Rare case of failure by an automated system to detect extended spectrum beta-lactamase in a cephalosporin-resistant Klebsiella pneumoniae isolate. J. Clin. Microbial. 37,2388. 62. Gerlich D., Poirel L., Lilleporn AETL. Molecular epidemiology of the integrome-located VEB1 extended spectrum beta-lactamase in nosocomial enterobacterial isolates in Bangkok, Thailand. J. Clin. Microbial. 39,175-182. 63. Patterson D.L. Resistance in gram-negative bacteria, Enterobacteriaceae. AMJ Med.119 S20 S28. 64. Patterson D.L., Bonomo R.A. Extended spectrum beta-lactamases, a clinical update. Clin. Microbial. Revelation 18,657-686. 65. Zhang X, NIY, Zhang YETL. Outbreak of infection caused by Enterobacter cloacae producing the novel VEB3 beta-lactamase in China. J. Clin. Microbial. 43,826-831. 66. Ho PL, Shek RH, Chow KHETL. Detection and characterization of extended spectrum beta-lactamases among bloodstream isolates of Enterobacter species in Hong Kong, 2000-2002. J. Antimicrobe. Chem Other. 55,326-332. 67. Patout J.D., Lawpland K.B. Extended spectrum beta-lactamase producing Enterobacteriaceae, an emerging public health concern. Lancet Infect. DIS 8,159-166. 68. Poirel L., Patout J.D., Nordman P. Carbapenemesis, Molecular Diversity and Clinical Consequences. Future Microbial. 2,501-512. 69. Panagia T., Galani I., Suli M., Adamua P., Antoniadu A., Guyamarello H. Evaluation of Chrome agar trademark KPC for the detection of carbapenemus producing Enterobacteriaceae in rectal surveillance cultures. INTJ Antimicrobe. Agents 37,124-128. 70. Cohen Stuart J. Leverstein van Halma, Dutch Working Party on the Detection of Highly Resistant Microorganisms. Guideline for Phenotypic Screening and Confirmation of Carbapenemesis in Enterobacteriaceae. INTJ antimicrobe. Agents. 36,205-210. 71. Loae, Verrill R., Williams J., Stratton C., Delalata P., Tang Y.W. Carbapenem resistance via the black APC2 gene in Enterobacter cloacae blood culture isolate. South. Med J.103,453-454. 72. Bush K., Jacoby G.A. Updated functional classification of beta-lactamases. Antimicrobe. Agents. Chem other. 54,969-976. 73. Nordman P., Nas T., Poyer L. Global spread of carbapenemus producing Enterobacteriaceae. Emergency. Infect. DIS 17,1791-1798. 74. Nas T., Nordman P. Analysis of a carbapenem hydrolyzing class A beta-lactamase from Enterobacter cloacae and of its LYSR type regulatory protein. PROC. Nat Academic. Sci. USA 91,7693-7697. 75. Radis M., Power P., Gutkind GETL. First class A carbapenemus isolated from Enterobacteriaceae in Argentina. Antimicrobe. Agents. Chem other. 48,1068-1069. 76. Potomarthy S., Molland E.S., Jurechko S., Swansea S.R., Thompson K.S., Fritsch T.R. NMCA carbapenem hydrolyzing enzyme in Enterobacter cloacae in North America. Emergency. Infect. DIS 9,999-1002. 77. Nas T., Cato N. C., Bernice S., Cousin G., Nordman P. First identification of BLA-IMI1 in an Enterobacter cloacae clinical isolate from France. Antimicrobe. Agents. Chem other. 56,1664-1665. 78. Rasmussen B.A., Bush K., Keeney D.E.T.L. Characterization of IMI1 beta-lactamase, a class A carbapenem hydrolyzing enzyme from Enterobacter cloacae. Antimicrobe. Agents. Chem other. 40,2080-2086. 79. Yun Song Y., Xiaoxing D., Zhihui Z., Yagang C., Lanwan L. First isolation of BLA-IMI2 in an Enterobacter cloacae clinical isolate from China. Antimicrobe. Agents. Chem other. 50,1610-1611. 80. Nordman P., Mariota S., Nas T., Labia R., Nicholas M. H. Biochemical properties of a carbapenem hydrolyzing beta-lactamase from Enterobacter cloacae and cloning of the gene into Escherichia coli. Antimicrobe. Agents. Chem other. 37,939-946. 81. Queenan A.M., Bush K. Carbapenemesis, the versatile beta-lactamases. Clin. Microbial. Revelation 20,444-58. Focuses on updated information on the epidemiological and biochemical characteristics of carbapenemesis of E. cloacae. 82. Aubryn C., Poirel L., Ashar J., Nordman P. Carbapenemus producing Enterobacteriaceae, U.S. rivers. Emergency. Infect. DIS 11,260-264. 83. Gaiacau pp. Psovlicus ls. Sacres a. Lacova v. Saphiano d. Salepi eibc1, a novel integron-associated class A beta-lactamase with extended spectrum properties produced by an clinical strain. Antimicrobe. Agents Chem other. 44, 2247 2253. 84. Bratu S., Landman D., Alam M., Tolentino E., Quayle J. Detection of KPC carbapenem hydrolyzing enzymes in Enterobacter species from Brooklyn, New York. Antimicrobe. Agents Chem other. 49, 776 778. 85. Corniglia G., Guaiamarello H., Rossolini GM. Metallobeta-lactamases, a last frontier for beta-lactams? Lancet Infect. DIS 11,381 393. 86. Deshpande LM, Jones RN, Fritsch TR, Sater HS. Occurrence and characterization of carbapenemus producing Enterobacteriaceae, report from the Sentry Antimicrobial Surveillance Program. Microbe. Drug resist. 12, 223 230. 87. Yan JJ, KOWC, Chuang CL, Wu JJ. Metallobeta-lactamase producing Enterobacteriaceae isolates in a university hospital in Taiwan. Prevalence of IMP8 in Enterobacter cloacae and first identification of VIM2 in Citrobacter freundii. J. Antimicrobe. Chem other. 50, 503 511. 88. Li MF, Peng CF, HSUHJ, Chen Yat. Molecular characterization of the metallobeta-lactamase genes in imipenem-resistant gram-negative bacteria from a university hospital in southern Taiwan. INTJ antimicrobe. Agents. 32, 475 480. 89. Lazaro F., Dockier JD, Colinon CETL. Emergence in Klebsiella pneumoniae and Enterobacter cloacae clinical isolates of the VIM4 metallobeta-lactamase encoded by a conjugative plasmid. Antimicrobe. Agents Chem other. 48, 648 650. 90. Perilli MG, Mesotesta ML, Marco FETL. Class I integron-borne BLA-VIM1 carbapenemus in a strain of Enterobacter cloacae responsible for a case of fatal pneumonia. Microbe. Drug resist. 14, 45 47. 91. Falcone M., Mesotesta ML, Perilli MGETL. Infections with VIM1 metallobeta-lactamase producing Enterobacter cloacae and their correlation with clinical outcome. J. Clin. Microbial. 47 35 14 35 19. 92. Panopolo M., Aelpopolo E., Economides A., Grapsa A., Paspalido E., Cardilectinidu S. Emergence of VIM12 in Enterobacter cloacae. J. Clin. Microbial. 48 34 14 34 15. 93. Suli M., Contopidu FV, Papatomycelaces E., Galani I., Armagonides A., Guillemot clinical experience of serious infections caused by Enterobacteriaceae producing Vim1 metallobeta-lactamase in a Greek university hospital. Clin. Infect. DIS 46,847-854. 94. Tato M. Koch trademark, Ruiz Garbajosa PETL. Complex clonal and plasmid epidemiology in the first outbreak of Enterobacteriaceae infection involving Vim1 metallobeta-lactamase in Spain, toward endemicity. Clin. Infect. DIS 45,1171-1178. 95. Yang D. Tolman Ma, Giska CGETL. Characterization of a new metallobeta-lactamase gene, BLA-NDM1, and a novel erythromycin esterase gene carried on a unique genetic structure in Klebsiella pneumoniae sequence type 14 from India. Antimicrobe. Agents Chem Other. 53, 50 46 50 54. 96. Brink A.J., Cutsy J., Clay CGETL. Emergence of New Delhi metallobeta-lactamase and Klebsiella pneumoniae carbapenemus in South Africa. J. Clin. Microbial. 50, 525 527. 97. Bogaerts P., Boucheruf W., Resende de Castro RETL. Emergence of NDM1 producing Enterobacteriaceae in Belgium. Antimicrobe. Agents Chem Other. 55 30 36 30 38. 98. Cara A., Poirel L., Yilmaz METL. Spread of OXA48 encoding plasmid in Turkey and beyond. Antimicrobe. Agents Chem Other. 54, 1369 1373. 99. Poirel L., Castanheira M., Cara AETL. OXA 163, an OXA48 related class D beta-lactamase with extended activity toward expanded spectrum cephalosporins. Antimicrobe. Agents Chem Other. 55, 25 46 25 51. 100. Glupchinskia Y., Huanga TD, Boucherufa WETL. Rapid emergence and spread of OXA48 producing carbapenem resistant Enterobacteriaceae isolates in Belgian hospitals. INTJ antimicrobe. Agents 39, 168 172. 101. Poirel L., ROSA, Cara AETL. Cross-border transmission of OXA48 producing Enterobacter cloacae from Morocco to France. J. Antimicrobe. Chem Other. 66, 1181 1182. 102. Sabo D., Silvera F., Hooger AMETL. Outer membrane protein changes and efflux pump expression together may confer resistance to ertapenem in Enterobacter cloacae. Antimicrobe. Agents Chem Other. 50, 28 33 28 35. 103. Chow JW, Fine MJ, Schloss DMETL. Enterobacter bacteremia. Clinical features and emergence of antibiotic resistance during therapy. N. Intern. Med. 115, 585 590. 104. Choi SH, Lee JE, Park SJETL. Emergence of antibiotic resistance during therapy for infections caused by Enterobacteriaceae producing AMP-C beta-lactamase implications for antibiotic use. Antimicrobe. Agents Chem Other. 52,995 1000. 105. Bach Aron S, Imberex H, Schossless Dankla E, Cloacae A. The ACRB multi-drug transporter plays a major role in high-level fluoroquinolone resistance in Salmonella enterica cerevar tifimurium phage type DT204 microbe. Drug resist 0.8, 281-289. 106. Ruiz J. Mechanisms of resistance to quinolones, target alterations, decreased accumulation, and DNA gyrase protection. J. Antimicrobe. Chem Other. 51, 1109 1117. 107. Perichon B, Curvalin P, Gallium-ANDM transferable resistance to aminoglycosides by methylation of G1405 and 16S rRNA and to hydrophilic fluoroquinolones by kappa-mediated efflux in Escherichia coli. Antimicrobe. Agents Chem Other. 51, 2464-2469. 108. Canomi, Rodriguez Martinez J.M., Aguero J.E.T.L. Detection of plasmid-mediated quinolone resistance genes in clinical isolates of Enterobacter species in Spain. J. Clin. Microbial. 47, 2033-2039. 109. Robixec A, Jacobi G.A., Hooper D.C. The worldwide emergence of plasmid-mediated quinolone resistance. Lancet Infect. DIS 6,629-640. 110. Martinez L, Canomi, Rodriguez Martinez J.M., Calvo J., Pascual A. Plasmid-mediated quinolone resistance. Expert. Rev. Anti-infect. There. 6,685 711. 111. Park Ch, Robixec A, Jacobi G.A., Sam D., Hooper D.C. Prevalence in the United States of AACIBCR encoding a ciprofloxacin modifying enzyme. Antimicrobe. Agents Chem Other. 50 39 53 39 55. 112. Jacobi G.A., Chow N., Waits K.B. Prevalence of plasmid-mediated quinolone resistance. Antimicrobe. Agents Chem Other. 47, 559 562. 113. Park Y.J., UJK, Lee S., OEJ, Wu G.J. Prevalence and diversity of QNR alleles in AMP-producing Enterobacter cloacae, Enterobacter aerogenes, Citrobacter freundii, and Serratia marcescens, a multi-center study from Korea. J. Antimicrobe. Chem Other. 60, 868 871. 114. Nordman P., Poirel L. Emergence of plasmid-mediated resistance to quinolones in Enterobacteriaceae. J. Antimicrobe. Chem Other. 56, 463 469. 115. Wu J.J., KOWC, Tsai SH, Yan J.J. Prevalence of plasmid-mediated quinolone resistance determinants QNRA, QNRB, and QNRS among clinical isolates of Enterobacter cloacae in a Taiwanese hospital. Antimicrobe. Agents Chem Other. 51, 12 23 12 27. 116. Khmeletsky I, Navonvenezia S., Strahilowitz J., Carmely Y. Plasmid-mediated QNRB2 and carbapenemus gene black APC2 carried on the same plasmid in carbapenem-resistant ciprofloxacin-susceptible Enterobacter cloacae isolates. Antimicrobe. Agents Chem other. 52, 2962 2965. 117. Jacobi G, Catwar V, Hooper DETALQNR. Gene nomenclature. Antimicrobe. Agents Chem other. 52, 2297 2299. 118. Neonacus I, Gaikas A, Cloacae, Manios A, Georgiladakis A, Silentis Y. Evolution of aminoglycoside resistance phenotypes of 4 gram-negative bacteria, an 8-year survey in a university hospital in Greece. INTJ antimicrobe. Agents 22, 526 531. 119. Kim Sy, Park Yj, UJK, Kim Ys, Han K. Prevalence and characteristics of AACIBCR in AMP-producing Enterobacter cloacae, Citrobacter freundii, and Serratia marcescens, a multi-center study from Korea. Dian. Microbial. Infect. DIS 63, 314 318. 120. Galani I, Suli M, Chris Suli Z, Orlando K, Guiamarello H. Characterization of a new INTE GRON containing BLAVIM1 and AACIC in an Enterobacter cloacae clinical isolate from Greece. J. Antimicrobe. Chem other. 55, 634 638. 121. Xavier B, Dowsicki M.J. Antimicrobial susceptibility among gram-negative isolates collected from intensive care units in North America, Europe, the Asia-Pacific Rim, Latin America, the Middle East, and Africa between 2004 and 2009 as part of the tigecycline evaluation and surveillance trial. Clin. 34, 124 137. 122. Anthony K.B., Fishman No., Lincoln Dr., Gasink L.B., Edelstein Ph., Lautenbach E. Clinical and microbiological outcomes of serious infections with multi-drug-resistant gram-negative organisms treated with tigecycline. Clin. Infect. DIS 46, 567 570. 123. Keeney D., Rusin A., Bradford Pa., Rama, a transcriptional regulator, an ACRAB, an R&D type efflux pump, are associated with decreased susceptibility to tigecycline in Enterobacter cloacae. Microbe. Drug resist. 13, 16. 124. Doral C., Fiant A.L., Bremont S., Courvalin P., Leclerc R. Emergence of an Enterobacter hormicii strain with reduced susceptibility to tigecycline under tigecycline therapy. Antimicrobe. Agents Chem other. 53, 49 53 49 54. 125. Phyllogasmi, Kasiaka S.K. Colistin, the revival of polymyxins for the management of multi-drug-resistant gram-negative bacterial infections. Clin. Infect. DIS 40, 1333 1341. Iratum in, Clin. Infect. DIS 42, 1819. 126. Price DJE, Gram D. Effect of large doses of colistin sulfamethate on renal function. BMJ 4, 525 527. 127. Cook-Weiser J., Seidel VW, Fetterman EB, Canarek P., Feiner DC, Eden AE. Adverse effects of sodium colistin methate, manifestations and specific reaction rates during 317 courses of therapy. N. Intern. Med.72,857,868. 128. Lee J., Nation RL, Milne RW, Turnage JD, Coulthard K. Evaluation of colistin as an agent against multi-resistant gram-negative bacteria. INTJ antimicrobe. Agents 25,1125. 129. Plachauras D., Carvanen M., Friberg LEET al. Population pharmacokinetic analysis of colistin methane sulfonate and colistin after intravenous administration in critically ill patients with infections caused by gram-negative bacteria. Antimicrobe. Agents Chem Other. 53 34 30 34 36. 130. Groisman E.A., Kaiser J., Sonsini F.C. Regulation of polymyxin resistance and adaptation to low Mg2+ environments. J. Bacterial. 179, 70 40 70 45. 131. Low Tenfo Jr., Desmet A.M., Didaren B.M., Klootmans J.A., Van Keulen Ph. Comparative evaluation of the Vitek 2, disc diffusion, test, broth microdilution, and agar dilution susceptibility testing methods for colistin in clinical isolates, including hetero-resistant Enterobacter cloacae and Acinetobacter baumannii strains. Antimicrobe. Agents Chem Other. 51 37 26 37 30. 132. Tassini C., Urbani L., Biancofiore Get al. Colistin in combination with rifampin and imipenem for treating a BLAVim1 metallobeta-lactamase producing Enterobacter cloacae disseminated infection in a liver transplant patient. Minerva Anesthesia al. 74, 47 49. 133. Centers for Disease Control and Prevention. Guidance for control of infections with carbapenem-resistant or carbapenemus-producing Enterobacteriaceae in acute care facilities. Morb. Mortal. WKLY Republic 58, 256 260. 134. Lucet J.C., Decray D., Fischella A.et al. Control of a prolonged outbreak of extended-spectrum beta-lactamase-producing Enterobacteriaceae in a university hospital. Clin. Infect. DIS 29, 14 11 14 18. 135. Samra Z., Beher J., Matter Shapiro L., Aziz N., Israel S., Bishara J. Evaluation of Chrome Agar KPC for rapid detection of carbapenem-resistant Enterobacteriaceae. J. Clin. Microbial. 46 31 10 31 11. 136. Ravi Kumar P. GenBank new holotype for Enterobacter cloacae subsp. Cloacae strain PR4 isolated and identified by 16S rDNA gene sequence with phylogenetic tree view, from explosive-laden soil. Int J. of res.in and applied Sci. 653 to 65 2016. 137. Paddy, S.K., Gokula, S., 2016. Benzene biodegradation by indigenous mixed microbial culture, kinetic modeling and process optimization. International Biodeterioration and Biodegradation, https://dx.doi.org/10.1016/j.ibiad.2016.10.011